Bruce Beutler is Regental Professor and Director of the Center for Genetics and Host Defense at the University of Texas Southwestern Medical Center in Dallas, and holds the Raymond and Ellen Willey Distinguished Chair in Cancer Research in honor of Laverne and Raymond Willey Sr. Born in Chicago in 1957, actually there's a slide here. And if you want to read more details, those who read the Journal of Clinical Investigation, there's a wonderful interview by Ushma Nil of Giants in Medicine. It appears in every issue. One of the earliest ones was Ernest Beutler. It's an MP3. You can hear it, and you can't obviously read it on the screen here, but it's well worth reading. Born in Chicago in 1957, one of Professor Beutler's earliest memories was the discovery of newly fallen snow and wondering who did that. Questions like that at such an early age were not surprising in his family, which upheld strong academic standards. As we've heard about his parents, his mother, Bonnie, was born in the USA to immigrants from the Ukraine. She worked for several years as a technical writer. Inquiry and discovery was instilled in each of the Beutler children. Bruce Beutler and two of his siblings became physicians, and the other sibling became a successful software designer and business person. Professor Beutler recalls a deep fascination with biology already when he was in the second grade. All he wanted to be was a biologist. He began working in his father's laboratory at the age of 14, learning to purify proteins and to characterize erythrocyte enzymes. Professor Beutler also worked in the laboratory of Susumu Ono, a mammalian geneticist famous for recognizing the essential role of gene duplication in evolution. Skipping several grades, Professor Beutler graduated from UCSD at the age of 17, I believe. He can correct me on some of these details. Following the counsel of his father, he went on to medical school at the University of Chicago, where he delved into the big challenges of biomedical discovery. He pursued a couple of years of clinical work, including neurology, in Texas at Southwestern, where he returned. Armed with a deep understanding and broad experience, he was now prepared to tackle any problem he chose. His achievements are proof that medicine and scientific research are not mutually exclusive, but actually intertwined necessarily for achieving advancement for the benefit of humankind. Professor Beutler discovered the key elements whereby infection is sensed, together with the major weapons in the arsenal of the innate immune system. Many of the molecules discovered and characterized are also targets for treatment of common inflammatory conditions. His discoveries have led to the development of new drugs for a wide range of diseases, including inflammatory bowel disease and arthritis. For these discoveries, Professor Beutler received a variety of awards, many of them, and recognitions, including the Shaw Prize, and shortly thereafter, he was jointly awarded the Nobel Prize in Medicine in 2011. Many people, after receiving a Nobel Prize, rest on their laurels, but not so for Professor Beutler. He has continued to break new ground in biomedical discovery, as we'll hear. A member of the National Academy of Sciences, Professor Beutler was recently awarded the 2013 American Society for Clinical Investigation Stanley Korsmeyer Award in recognition of his contribution to the field of innate immunity and his continued leadership and continued scientific contributions since his seminal work on mammalian toll-like receptors. Professor Beutler continues to address medical challenges through scientific research at all levels, ranging from molecules to organismal models and human studies. His agreement to accept the Korsmeyer Award of the ASCI and his support for today's symposium once again emphasized the great importance of basic research to the advancement of clinical medicine. Like his parents and family, Bruce is a true and great friend of Israel and of Israeli science. I had the unique personal privilege of visiting Bruce in his research office at Scripps in August 2011, just a few short months before he won the Nobel Prize in 2011, and just before his return to Dallas. I'd just like to make 2 points about that visit, during which I gave a one-on-one research presentation. First is Bruce's modesty. As I was trying to convey certain hypotheses, I had no idea of the depth and extent of his expertise, and that I was having the opportunity of a lifetime, and Bruce listened, very engaged and very patient. Second, one of his clear-cut recommendations was to generate organismal models as a key step in continuing the research, and of course it turns out that he was correct. This also fits with the recent breakthrough successes which Professor Beutler will share with us today in his presentation, entitled Monitored Mutagenesis of the Mouse Genome and High-Speed Identification of Mutations that Cause Phenotypes. So it's really a great honor, privilege, and pleasure to welcome Bruce to the podium.
Well, thank you all. There are a lot of you, I see. It's really a pleasure and an honor for me to be here. I think it's my 6th trip to Israel. I've loved every trip that I made here, and it's especially sweet to come here for this purpose, to help commemorate the contributions of my father and the generosity of my mother. And I would like to start with a slide of them that people rarely see. This was the couple when they were in their early 30s. I don't know the exact year this picture was taken, but I know that I took it. It was at our then relatively new house in Arcadia, California. We had moved from Chicago just a short time earlier. My father looks very relaxed in this picture, but he was an energetic person, as you've heard. He was something of a polymath. Many people who work on so many different things turn out to be dilettantes, but that was definitely not the case with my father. He had a tremendous interest in all different topics in biology, ranging from glycolipid storage diseases to abnormalities of red cell enzymes, to cancer, especially leukemia and how it came about, to iron metabolism, and on and on. And he was a prolific writer. He wrote more than 1,000 papers during the course of his life. And as was mentioned, I received my first education in science from him. I worked in his lab when I was 14. I remember being set to work grinding placentas in order to extract glucocerebrosidase from them, and graduating soon thereafter to studying enzyme kinetics, learning how to do starch cell electrophoresis, learning how to isolate proteins, and I became extremely confident of my abilities, certainly way too confident, and it wasn't until a short time after I left the lab that I had my first bruising experiences with failure in science. But that didn't last. I was persistent, actually, just as my father was persistent and encouraged me to be the same way. My parents were, in my father's case, an immigrant. As you heard, he came from Germany. He was born there in 1928. My mother was born in the United States to immigrants from Ukraine. It was because of their backgrounds that they fell in love with Israel themselves, and they transmitted this to me, and I've passed it on to my own children, and so it's always a joy to visit here. I will talk to you today not so much about the end product of our work, although I'll tell you our objectives and what we hope to achieve, but I will tell you about the technology and how it's changed. And I might even modify the title from high speed to instantaneous identification of mutations, because that's what we've been able to achieve. It sounds a bit magical, but I'll tell you precisely how it's done. The problem that we started with many years ago, the interest that I have ever since my father encouraged me to read The Microbe Hunters by Paul de Kruijff, is the problem of infectious disease. It's a fact that about 1 out of every 4 people born will ultimately die of an infection if one looks worldwide. And of course, it's not nearly so much of a problem in developed countries, but all the world was the developing world a short time ago. We have to always keep that in mind. And if we look only at the developing world, then many more people die of infection. A much higher percentage will end up dying of infection. And furthermore, infection takes life very early on. It's mostly children who die of infection. One could say that infection takes more years of human life than any other cause of death. Because infection is something that's largely restricted to the years prior to and during reproduction, it's an enormous selective pressure in our species. And it may have been so always among Homo sapiens. It may be true all throughout the tree of life, at least going very far back. And so we might imagine that many genes have arisen de novo or have been modified to help us cope with infection. And we know this is so in specific instances, and I won't go into them one by one. I'll just say that it was infection that impelled the evolution of the immune system. Both the innate and the adaptive components of immunity came into being because of the pressure that fungi and parasites and bacteria and viruses represent. Probably there are thousands of genes necessary for comprehensive immunity, and as of this day, we still don't have a complete list of all those genes. We do know that failure of immunity in one way or another is not rare. And there are hundreds of immunodeficiency disorders cataloged already, but certainly not all of them are known, nor do we have a really vivid picture, at least to my mind, of exactly how immunity operates. We draw pictures, we have general schemes in mind, but I would argue that we don't know the half of it, nor the 10th part of it, nor probably 1% of everything that really happens to combat an infectious organism. The flip side of immunity is well known to you also. Because we have an immune system, we're at a certain risk to develop autoimmune disease. Having an immune system is a dangerous thing, in effect. And all of the inflammatory diseases we know, virtually everything that ends with itis, we can say came into being as well as a consequence of infection. So here I show illustrations of rheumatoid arthritis, gout, also lupus at the lower right, and type 1 diabetes. All of these diseases come about because we developed a means to fight infection. And the methods that we have to contain the immune system, to guide it properly, are imperfect, or at least they fail in some people. I tend to think of the immune system as a machine distributed over many different kinds of cells, having thousands of protein components, as I mentioned. Certainly it's something much more complicated than this watch. And to my thinking, the first step in understanding how a machine works in real detail is to have a list of the essential parts. And the question is, how can we acquire a list of all of these parts, all the parts that are truly essential for function? One way to go about it is to take the unbiased approach of random germline mutagenesis, use a powerful mutagen in whatever model organism you wish, destroy the genome piecemeal, a few genes at a time in each organism, look for exceptions to the norm in immunity, and ultimately you can put together a list of parts, and then you have the problem of understanding how they fit together. You have to reassemble the watch, and that requires usually other tools, but it's definitely a starting point. Forward genetics, then, is that process by which you keep a phenomenon, in our case immunity, under surveillance. You modify genes a few at a time, and you look for phenotypes. And when all genes have been heavily mutagenized, then you can be quite confident that you have a list of the genes that are essential for a particular phenomenon. This is akin to reverse engineering, as engineers do. You try to understand how the living system works by making it fail over and over and over again in different ways. In mammals, which is what we like to work with, the tool of choice for generating phenovariants is a chemical called ethyl nitrosourea. And all of the mice that you see here, whether they have behavioral abnormalities or visible abnormalities, in fact only one of them, the black mouse at the lower left, is of the reference strain with a defined sequence of nucleotides. All of them are abnormal because of a single change, a single nucleotide change somewhere in the genome. And the idea then is to make such variants, to keep immunity under surveillance and to track the variants down and figure out exactly what's wrong. This process begins when one injects a male mouse with ethyl nitrosourea. It's a fairly simple molecule, as you see, and it's given in 3 divided doses of 100 milligrams per kilo of body weight, and a period of infertility then ensues, during which the pool of spermatogonia is heavily depleted. Perhaps only 10 to 100 spermatogonia survive, and then they grow back and recolonize the testis, and they're heavily mutagenized, and from then on, the sperm that are produced by that mouse, which we call the G0, have a heavy burden of mutations. How heavy is that burden? Well, actually, we know within rather precise limits how much mutation we create in every sperm. We know that there are an average of 134 mutations per spermatogonium that cause coding change, and we also know that it's almost always the case, if you see a phenotype, that it comes from a change in coding sense or a change in splicing, let's say, not from intergenic mutations, not from mutations that affect promoter sequences. I'll come back to that number and how we know it, but I'll say that, of course, half of these mutations are thrown away in the process of meiosis, and half of them are packaged in a sperm. So to our estimate, 67 mutations ultimately are passed on to the G1 mouse on average, and these may or may not result in a phenotype, an immune phenotype that we're interested in. Typically, we combine two G0 mice into a single pedigree to concentrate mutations. So we generate the G2 mice, finally the G3 mice, which we screen for phenotype, and we can calculate on average there'd be about 68 mutations per G3 animal, and 11 would be homozygous. And these are mutations that affect coding sense. And I mention again, the G1 is very important. It's that mouse that we go to determine just what mutations are present. This used to be a blind process, you see. We didn't know what was there, and we would screen the G3s hoping that there would be mutations. But nowadays, with the G1s, we do two things. First of all, we harvest their sperm, and we archive them in a frozen state so we can recall the mutations anytime we want. And we do whole exome sequencing to determine just what the mutations are. This is done nowadays on a HiSeq 2500 machine, and this machine will deliver about 1 trillion base pairs of sequence every 3 weeks. The throughput then is 0.6 trillion base pairs per run of 11 days. We define the exome rather generously as being 52 million base pairs in size, which accounts for all coding sequence and for quite a bit of flanking sequence on either side of every exon. We can run about 80 G1 mice, or 80 exomes per run, and we get coverage to a depth of about 140 reads per base on average, and that assures that greater than 96% of the target is covered to a depth of 10 or more, and that in turn means that we'll discover point mutations with about that efficiency. It's not 100%, but it isn't bad. Most of the time, if we should see a phenotype, we know that the mutation that causes it will be included in that sample. How do we know so accurately the number of mutations per sperm? Because by this time, we've done actually well over 1,000 whole exomes from G1 mice, and this is the distribution of the number of mutations per exome and the number of exomes that had that number of mutations shown in windows of 5. The peak value falls, I think, it looks to me like at 65 to 70, and the absolute mean is 67. In the double pedigrees that I constructed, which amount to about a third of the pedigrees we make, the average number of mutations per G1 is 91 coding changes. At this point, the mice are screened for phenovariants, and we look for exceptions both in terms of visible mutations, which we can't bear to just throw away, and also in terms of immune performance. And there we have a series of screens. We look at macrophage responses to various inducing conditions. Those are a set of innate immune screens that probe the toll-like receptor signaling pathways, the inflammasome, also systems for double-strand DNA sensing, and so on. We have adaptive immune screens which measure the ability of mice to make antibody responses, both to T-dependent and T-independent challenges. We look also for the development of immune cells, in fact all hematopoietic cells in the blood. We look at GI homeostasis. What does it take to maintain a non-inflammatory state in mice that have been challenged with DSS? And we also ask whether the mouse can survive a normally harmless viral infection. This is mouse cytomegalovirus challenge. Most mice are unharmed by that. The wild type will never die of it at the dose we give, and exceptional mice will die of the infection if they have critical mutations. We've been at this for several years now, actually for about 14 years now, mostly at Scripps and some at UT Southwestern. As I mentioned, the intention has always been to create phenotype, then to solve it by positional cloning. I also mentioned the process used to be blind. We didn't know how much damage we were doing to the genome, either in a single mouse or in large collections of mice that we'd exposed to the mutagen. We just sort of had to guess about that. Nonetheless, we went ahead and we did create hundreds of phenotypes and solve hundreds of phenotypes over a decade or so at TSRI. And this process certainly began to accelerate as soon as the reference genome of the mouse was published in 2002, whereafter it was no longer necessary to go searching for the gene content of a particular interval. And it became faster again when massively parallel sequencing technologies like HiSeq became available. Then all the candidate mutations were known up front, and we could also begin to do probabilistic calculations of saturation. Here's one such example. In this slide, we see what the computer says about how much of the genome has been damaged in a population of 13,750 G3 mice that were screened for a particular phenotype. It doesn't matter what that phenotype is, of course. We're asking how many genes were damaged, how many overtly destroyed by premature stop codons or critical splice junction errors, how many probably damaged according to estimates by programs like Polyphen2, how many simply touched. And the answers are that when one looks at this collection of mice, the computer will recreate all of the pedigrees and look at all of the mutations and calculate the likelihood that they were transmitted to homozygosity, and it will add up the likelihood that each individual gene was damaged. You can see that about 2,000 genes were mutated to a probable null status, and about 1,613 were probably transmitted to homozygosity, and so that would be something like 7% of all genes that were clearly overtly destroyed. But it's only an estimate, and we don't pay attention in this case to how many genes are homozygous lethal and never showed up, for example. It's just the best that we could do at that time. We began along the way to archive mutations, as I mentioned, and at this date, we have 98,553 coding changes stored away in frozen sperm, and they affect 83.5% of all protein encoding genes. We have clear null alleles that affect about 22% of all genes stored away in sperm, but probably about 35% have been mutated to a state of phenovariance. We know that more phenotype comes out of missense alleles than null alleles just because they're more abundant. And most of this was done just over the past year. We aim to go to close to 1 million mutations, and we should at that point have damaged by far the greater part of all genes in the mouse genome. But while this was going on, a growing problem began to present itself. We found that the production of phenotype and the identification of mutations were growing mismatched, maybe because we were overconfident about how to find them. Historically, we would look for a phenotype. If we found one, we would expand that pedigree. We'd back-cross the mouse to the founder G1, try to make more of them, make a homozygous stock, outcross, back-cross, map, and then find what the causative mutation was. This was a slow process, and it went on for quite a while, and the number of putative mutations began to greatly exceed the number of mutations that actually were found. In fact, there always has to be such a gap because you don't find the mutation instantly, at least in the old days you didn't, and we found that this gap was getting larger and larger. As of recently, in January, I cataloged all the phenotypes we'd ever pointed to, and we had 618 of them. Of those, 289 had been mapped, and 278 had been found, and they fell into 188 genes. So most of them remained unfound. For immunologic phenotypes, the figure was 412 in total, and we had mapped 154 and found 147. So clearly there was a great lag between finding a phenotype and being able to actually map it, and some of them proved to never be mappable for reasons we didn't understand. A good example of how things were going in one specific screen would be provided by a screen for the antibody response as well as the developmental screen by flow cytometry to look for defects of immunity. This was a fairly elaborate screen that involved 3 challenges with 2 T-dependent antigens and 1 T-independent antigen, and then bleeding the mouse and looking for antibody titers against all three of those antigens, and looking also for cellular defects in the peripheral blood. In all, something like 13,570 mice had been examined in the first of these screens, the most abundant of them, and one of them was started rather more recently, and only 3,876 animals had been tested. Where mutations that impaired the antibody response were concerned, we could break them down into three colors, into the T-dependent screens for response to rSFV-bgal, recombinant Semliki forest virus, expressing beta-galactosidase, or into ova-alum, shown in blue. Those were both T-dependent screens, and then there was a screen for T-independent challenge with NP-ficol, which is shown in green, and there would be overlap between them. Some mutations would cripple all of these responses, some were specific to one. But of all the phenotypes that we named there, only those shown with black dots were actually found, and that amounted to 41 causative mutations identified in a total of 39 genes. If we took into account the flow cytometry assays, we found something similar. We found a total of 64 causative mutations in 56 genes. So something close to 75% of the mutations we simply weren't able to track down, at least in a timely manner, and this began to greatly bother me. So the situation was only 26% of the phenotypes had been solved. We were producing phenotype much faster than we could find it, and I began to dream of a way to find the cause of phenotype much faster. And I imagined, wouldn't it be nice if we had something akin to Google glasses as molecular geneticists, if we could simply gaze at a collection of mice like this one, and the glasses would point out to us which mice really had a variant phenotype. Of course, it might be obvious, as in this cage of mice, you see several of them are blotchy. Given that you could see which ones had the phenotype, wouldn't it be even nicer if you could know just instantly, in the blink of an eye, what gene had been hit among those 91 or so that was causative, and whether it was new, and actually what it did to the protein, and so on and so forth. And then it occurred to me all at once that we could do that. No problem at all. We could do that, and I'll tell you how we do it. We have devised now a way to find the cause of phenotype in real time. And it works like this. When a phenotype is observed, the cause is now usually known at the same instant, or else the phenotype is rejected as being non-genetic. And this applies to all phenotypes that we might study, whether they're visible or behavioral phenotypes or defects of the immune system. It applies to qualitative phenotypes like black mice versus white mice, or to quantitative phenotypes like the antibody response being only 25% what it should be. And there are a few exceptions, which I'll come to, which are very interesting in and of themselves and lead us into the question of complex phenotypes. Nowadays, what we do is we take all G1 males, as I mentioned before, we isolate and archive their sperm, and we also sequence their whole exome. But the new step is that at the moment we know all of the mutations present in the G1 male. At that very time, we order an AmpliSeq panel that enables us to genotype all of those mutations. Whether there are 120 of them or only 60 of them, it doesn't matter. The AmpliSeq technology is one in which you use in a single reaction vessel DNA from a given mouse and let's say 103 different primer pairs that are calculated not to interfere with each other, and typically all of the target sequences will be amplified at once. The G1 males are then bred to make 10 G2 females, and we make 50 G3 males if we can. And all of the G2 and G3 mice are genotyped at all of the mutated sites, which is a considerable amount of work, but it works well with the AmpliSeq technology. And all of this is done in advance of screening. and all of the data are uploaded to the computer and are waiting for the phenotypic data to arrive. As soon as the phenotypic data arrive, the computer will analyze the phenotypic data in light of genotype and will immediately declare whether a certain mutation is implicated. Nowadays, a human is no longer allowed to declare a phenotype in the lab. Only the computer is allowed to declare the phenotype, and that takes care of our problem with the gap. Either a phenotype is caused by a mutation, or as far as we can tell, it's not. Furthermore, the analysis is carried out by pedigree. In the past, we would just dispense mutagenized mice to postdocs and students in the lab to screen them, irrespective of whether they came from one pedigree or not. Now we withhold them until a pedigree is complete, and the pedigree is then dispensed for screening. So here is how our pedigrees look now. We have 50 G3 mice, 10 G2 animals. They come all from one G1, which has been harem bred to 4 B6 mice. The genotyping is carried out using ion torrent sequencing. There are several ways that it might be done, but this way is particularly reliable. The ion torrent is a benchtop instrument, which many of you might have seen, and it's capable of delivering about 1 billion base pairs of sequence, but in only 8 hours. And it's therefore capable of covering in a given mouse, or even in a couple of pedigrees, all of the mice at all of the mutant sites to a considerable depth. And this is how the data from the ion torrent look. You get a series of numbers where you have fractions that show the number of variant calls over the number of reference calls. Highlighted in blue would be a typical homozygous variant with 514 calls of the variant base over 0 of the wild type base. Then in yellow you see a typical wild type call of 0 variants over 323 reference. And next you see a heterozygote with about equal numbers of each. And when you have these kinds of numbers, you can be very confident about the genotype. But just to be sure that you can be confident and that I'm not just showing you the very best 3 genotypes, I'll show you some data. We are looking here at the results from 3 different pedigrees subjected to ion torrent analysis. So we have something like 10,000 genotypic calls in this histogram. And on the x-axis, we're looking at the percentage of variant calls in each of those genotypic calls. And I'll blow it up so you can see a little bit more clearly that we have a reference population at the left, a population of heterozygotes where you have near 50% of each base and variants. And what you should notice is that things go very close to the baseline here, so that if you exclude even a very few mice around the 80% or 20% mark, you have no ambiguous calls at all. You're genotyping perfectly. Let me show you how this works, first with a very simple example and then with a few more complicated ones. We happened upon a pedigree where some of the mice were small. About 9 out of 48 mice were small. We were still in the days when we allowed humans to name pedigrees, and so this was called the teeny phenotype, and it was subjected to analysis in just the way that I showed you. And here were the data that result. You can see arrayed along the top axis are a collection of all the G3 mice in the pedigree. Every column is a different mouse, and arrayed along the y-axis you see all of the candidate mutations, each in a different gene. And what I would like to point out is that we've colored all of the genotypic calls differently here. We've also noted that in some cases where there's red color here for the G2 female, the computer did a check and established that this G2 female wasn't really the mother of all of the G3 mice. It was not possible because she was not homozygous. She was not heterozygous for some of the mutations for which there was homozygosity in the G3 mouse. And so the computer picks up mistakes that are made in the vivarium. I'll tell you that doesn't matter at all as far as the assessment of linkage in this case, but it does matter when one is trying to track down synthetic lethals, which is something we can now do. In any case, the computer analyzes all of the genotypic calls, and in this case we're dealing with a simple qualitative phenotype, affected versus unaffected, and it declares that there is linkage on chromosome 1 to a mutation which is in a gene called KBTBD2, one of the BTB domain transcription factors, about which nothing has been published. Evidently, it's required for normal growth of the mouse, and we knew it just instantly. We didn't have to do mapping, essentially, because the mapping was already done up front. Now, in fact, we don't look only at one visible phenotype in a pedigree. Every pedigree is analyzed for 45 different phenotypes. You see a list of them here. In every case where we're talking about quantitative phenotypes, a graph showing the distribution of the phenotype is made, comparing wild type to variant. And the computer actually performs the analysis on every one of these phenotypes, but it does it all very quickly. Here would be an example of such a quantitative phenotype. We're looking here, and in the other examples I'll show you, at a failure of the antibody response to immunization with a recombinant Semliki forest virus vector that expresses beta-galactosidase. The C57 black 6 mice, shown on the left column, do respond to immunization, and every color that you see represents a different day, because in those days we weren't dispensing the pedigrees all together. They were done piecemeal on different days, and we would normalize the mutant data against the wild type data, and the wild type all come to an average of 100%, but you see right away in this pedigree there is something wrong with the response to this immunogen. And it doesn't look particularly Mendelian because, in fact, well, you'll see why. It's not 100% penetrant, and neither does it cause a completely qualitative effect. And suffice it to say, it would have been extremely difficult to map by the old methods. The G1 grandfather of this pedigree had 79 coding mutations. All of them were already known. There were 48 G3 mice to analyze, and that meant genotyping at 3,555 sites in all. And in this case, we undertook a continuous variable approach rather than simply saying that this was a qualitative trait, and we could have looked at just the very lowest 2 or 3 mice and at all the top mice. We looked at them all. And again, we arranged them into this kind of array, and all the data are analyzed computationally. What we see in this case is that there is linkage to a gene called AOX3, aldehyde oxidase 3, which is on chromosome 1 again. And in this case, the linkage appears to be principally recessive, but it does have a weak dominant component. We can look at the performance of mice with either homozygous variable or heterozygous or wild-type genotype, segregating them that way within the pedigree, and we see that all 4 homozygotes for this mutation are poor responders when immunized with the beta-gal encoding vector. The heterozygotes have intermediate performance, and those with the reference allele are mostly fairly normal, but not all of them are totally normal, which brings me to the next point, that at times, despite what we would like to think, ENU doesn't create only one mutation that affects performance in the assay we're looking at. Only after a phenotype is mapped in this way is it given a name now, and this was called AMBER. You might wonder why it is that aldehyde oxidase 3 has some effect on immune performance, and I'll tell you I don't really know the answer there. It's somewhat surprising. Of course, that's the best situation. We like to find things that are new and surprising. We could imagine that aldehyde oxidase 3 prevents cross-linking of DNA by aldehydes. We know that Fanconi anemia mutations, which create some immune disturbance, work their effect by preventing the removal of such cross-links. And there are obvious ways to investigate just what's going on in this particular mutant. Sometimes ENU does cause complex phenotypes, and in former days we simply wouldn't have paid attention to them at all, and in fact I wasn't really aware that they occurred as often as they seemed to. And I'd like to tell you about now a pedigree called R0096, which was distributed like this. We're looking at the same sort of situation here with raw data. And we can also look at the normalized data. You see there's a population of low responders there, even going down to zero response. But again, it doesn't look particularly Mendelian. There are just too few of those mice to make good sense in terms of a single mutation, at least one that's consistent with full viability. When we do the linkage analysis now, we see that there's no linkage using either a recessive model or a semi-dominant or a dominant model. Nothing comes up to the Bonferroni line, which is the top of the two dashed lines. And so we have to wonder, where is this phenovariance coming from? Obviously not from just one site. A program was then written by Tao Wang in our biocomputation division to array all of the mutant sites against one another and to ask, is there a strong effect on phenotype if we take two mutations, if we only look at mice homozygous for two different mutations? And in fact, it looks also at semi-dominant or at dominant cases as well. The heat map that you see here corresponds to this color-coded key, and we do indeed find a pair of genes which, when both are homozygous, gives a very high Lod score of something like 10 to the 6th. Keep in mind that we're doing a lot of comparisons here, and the Bonferroni correction has to be pretty severe in order for us to interpret this. But when all is said and done, one would expect to find this kind of excursion from the expected only one in about 50 pedigrees, and I think it's very likely that it's true, though we're by no means sure of it. What are the two genes there? Well, you'll probably laugh when I tell you, because I can make no sense of it at all, but I'm telling you how these things are done and not trying to vouch for their truth at this point. One of the genes is cis, which is sucrase isomaltase, a brush border enzyme. The other is SICP2, which is a synaptonemal complex protein, also playing a role in mitosis. All I can say is that if indeed these two mutations in their homozygous form, when combined, will abolish this antibody response, it's something pretty surprising and remarkable. How do we come to be sure of that? Well, we have to go further. We have to knock out both of these genes or replace the mutation exactly in mice that have no other mutations from the pedigree and see if the phenomenon is reproduced. But what you can see is that when you have homozygous variant genotype at both of those loci, you have essentially no antibody response. And with either of them in less than a homozygous state, you do have an antibody response, and this is calculated to be highly significant, even allowing for the Bonferroni correction. At times, the complex traits can be of a kind of interfering type instead of mutually reinforcing. And I have one example of that. It's the last example I'll show. But a certain pedigree also showed weak performance in this particular screen, pedigree R0139. And there you see it with raw data, and here you see it normalized. In this case, what we observe in the recessive model of linkage is that one locus called NELL2, N-E-L-L2, slightly exceeds the Bonferroni threshold. But the linkage is still very weak, and it doesn't seem to begin to account for what we saw in the previous slide, for all of the variance that was observed. When we use an additive model, a semi-dominant model of linkage, then NELL2 is not quite as prominent. Another locus is prominent. This one is ATP2A2, which I'll return to in a moment. But I'll tell you that this variant is in the wrong direction. These mice actually have very high antibody performance, so high that the computer noticed them as an exception, and it ranks this as something significant in terms of linkage. And you see it even a little bit more strongly in a pure dominant model where NELL2 is excluded completely. And the immediate question is, could heterozygosity at this locus be interfering with homozygosity at the NELL2 locus? Could it be rescuing the mutation? And could that be why we find such weak effects when we look at linkage? Again, we can refer to the heat map and we look for inhibition of one locus by another. And this is an unbiased process. We simply run the program, and sure enough, the two genes that intersect and that give actually a fairly phenomenal score, something close to 10 to the 10th log value, are NEL2 and ATP2. It is the combination of those two that tends to weaken the appearance of linkage. We see that homozygosity for ATP2A2 mutation, in this case, this particular allele, is always lethal. None of the homozygotes show up anywhere in the pedigree. What we also see very clearly is that if we have homozygosity for NEL2 with a reference allele at ATP2A2, then we have underperformance in the antibody screen, quite significant underperformance. But if at the same time you have homozygosity for NEL2, you are heterozygous for the ATP2A2 allele, then there's rescue, there's normal performance again, and this is why there's such a weak appearance of linkage at the NEL2 location. ATP2A2 and NEL2 actually do have something to do with each other. ATP2A2 is a calcium channel that admits calcium to the endoplasmic or sarcoplasmic reticulum, and NEL2 is a calcium-binding protein, or a protein with many calcium-binding motifs that's secreted by way of the ER pathway. And one can make up stories of why one mutation would be likely to inhibit the other mutation, and those certainly need to be checked. Nonetheless, it seems very likely that we have picked up an authentic relationship in this case. Now, I want to close with just a few thoughts about what we're doing. There are really a lot of ways to do this. You're going to hear from Professor Skorecki a short time from now about practicing genetics, in a sense, in the natural world with human populations. I prefer mice in a way because one can do things with mice that one can't do with humans. Notably, you can breed them at will and make huge pedigrees to allow instant mapping of mutations, as I've shown you. But what we're doing is not so unnatural where humans are concerned. Chris Goodnow, a good colleague of mine who also does ENU mutagenesis, visited Scripps not long ago and said really exactly what I had been thinking for a long time, which is that we are all mutants. If you consider that each of us has one or two mutations of very recent origin passed down to us by our parents, and each of them had mutations passed down to them by grandparents, and you go back a mere 10 generations or about 200 years, you find that there's as much recent mutation in each of us as there is in an ENU mutagenized G1 mouse. And this is largely in heterozygous form. Dominant phenotypes with or without cooperativity may account for a great deal of human genetic variation. And I think it's something really well modeled by what we do with a chemical mutagen. Mutations don't occur in a vacuum, obviously, and mutations sometimes seem to be much more than the sum of their parts. And we're beginning to see this by looking at complex traits in the way that we are. There are about 23,000 genes, and if you took the total number of possible pairwise interactions between genes or between mutations in each gene, there would be about 264 million such interactions possible. These and the higher order interactions between proteins that we know exist are what make us more than the sum of our parts. They make us more than 23,000 functions happening separately from one another. Mutations at multiple loci clearly can interact, and perhaps this is one of the major secrets of evolution and why it seems to proceed so quickly, despite the fact that the mutation rate in nature is relatively low. With our system that I showed you, we can hope to test something a little bit under 1% of all pairwise interactions among genes each year, given an average of 70 coding splicing changes per pedigree. But when even one such interaction is found, if it's authentic, it's usually quite surprising, and it can be extremely informative. This is my group in Dallas. It's a large group. It's actually larger than that now, because I think a lot of people are attracted to this type of approach, to a pure exploratory approach that can yield surprise. And maybe some of you are as well. I hope so. I hope I lit some fires here, garnered some enthusiasm. And for those of you who are trainees, we're always looking for more people to join us. The immune screens that I showed you were done by these three people, Ming Zhang, Scott Wang, and Jin Choi. And they've worked their way now through, I think, about 15,000 mice. But the new standard will be about 1,000 per week, starting in April. And I want to thank my father once again, without whom this symposium wouldn't be taking place, and I would certainly never have gotten anywhere in science. And I want to thank all of you for your attention. Thank you.
I think that was one of the most exciting lectures we've had the privilege of hearing, and I thank Professor Beutler. It's an understatement for everything, and also for sharing with us a fresh new approach, which is rather remarkable. And he hinted that there was going to be this remarkable revelation. I don't know if it's been presented previously or elsewhere, so it's a tremendous privilege, and we thank you, Professor Beutler, for that. And we will consult with Professor Beyar. We do have, and we thank you also for leaving a little bit of time, if it's okay with you, for one or two questions. And I'd like to take the moderator's privilege with a question, because you said at the beginning, you confined your view in the ENU mutagenesis to genes and splicing sites and not intergenic regions. So, and given the high identification rate, what does this teach us about intergenic regions, so-called dark matter, and the large amount of effort that's being put on those intergenic regions? What would be your message?
Yeah, it's a really good question. I have to say, we're using a point mutagen. If we were using X-rays or an insertional mutagen, it might be a very different story. But I do think that there's quite a lot of redundancy within promoter regions, and if you destroy one critical element, that's not always enough to do it and produce a phenotype that you can find. Of course, as things get even cheaper, and we're now below $1,000 per genome, maybe someday we'll be at $10 per genome, and then of course we'll look at everything. And we'll have thousands of ENU mutations per pedigree to examine, but it should be feasible to track anything down, intergenic or not.
Thank you. Any questions from the audience? Yes, Mark, Professor Rothenberg.
Very elegant, outstanding work. I certainly learned a lot. I wanted to ask you, related and follow up to the question, is perhaps also your system, I guess, appears to not look for epigenetic modifiers. And I wonder if you could also comment about that, if you think you could potentially manipulate this system, if there's another way to actually do these types of experiments so you could look at chromosome involvement and epigenetic effects.
Occasionally, we do find mutations that are epigenetically influenced, let's put it that way. One example was the worker mutation. It was another mutation that affected B-cell performance, and so they were given names like that. These are mice that, the mutation is homozygous lethal. The heterozygotes have low penetrance of expression of the phenotype, and it was very difficult to track it down. The mutation is in rSFV-bgal2, I'll tell you, which is a protein involved, homologous to one involved in the UVB response in bacteria. And it's a part of a complex that associates with chromatin. And to make a long story short, mice that are affected and that have underperformance in their antibody response also have a fairly low CD4 T cell count and also have a white belly spot. And the concordance of the white belly spot with the immune phenotype is 100% in more than 100 affected mice seen, never one without the other. And the last time that melanoblasts and hematopoietic stem cells have a common origin is way, way early in development. I would say maybe at the zygote. So you have to imagine that this mutation affects a certain process that either must happen, to influence both of those lineages, and it either happens or it doesn't, and that process is probably epigenetic. So you can get at epigenetics through simple base pair mutation changes. On the other hand, if you're thinking of epigenetics in the classical way, people have done screens to look for that, and one can look at it, for example, a transgene that's variably expressed in a cell population like erythrocytes, and one can find mutations that shift the expression either to higher or lower levels, and usually they have global effects as well on coat coloration too, for instance, if you put it with the agouti hypervariable locus. So the complicated story boils down to the answer, yes, you can look at epigenetics this way.
Ruthie, Dr. Hirschberg, Professor Hirschberg.
So you mentioned that when you get a phenotype that you can't map to one of the mutations, you reject it as non-genetic. And I was wondering if you have any data on what proportion of mutations you do that for, because then that's the mutations that are basically not in the exomes or are epigenetic or are not. So.
Lately, we've been trying to close that gap that I talked about between something 600-something and 400-something mutations found. And what we are finding is that probably more than half of those that we have not found previously are unfindable, and the other half or so are findable by this approach. So I think a lot of those mutants that were selected by the human observer were a little bit wishful thinking, and they're non-genetic probably. At least we can't find them with our very best current technology.
Okay, so once again, on behalf of all of us, I'd like to thank Professor Beutler for this magnificent opportunity and these insights, which I think you wanted to provoke a lot of thinking and to give us a thrill of science, and I think you've succeeded very, very much so. And I'd like to now close the first part of the morning symposium with just saying thank you to everyone for coming, and you're welcome back, of course, and you should come back in 15 minutes after a coffee break, and there's coffee and refreshments outside for the second half. Thank you very much.